So I'm going to start with some disclosures. There are groups from which I derive some income or support, but I'm not going to be talking about any of that stuff today, nor will I be talking about investigational drugs or off-label uses. Okay, this is the face page of James Parkinson's 1817 monograph on Parkinson's disease. He didn't call it Parkinson's disease. He called it the shaking palsy. He's a really interesting character, and so I thought I'd share some background with him. Okay, who was he and who were his patients? He lived almost his entire life in an area of London called Hoxton Square, and that was it in the 1800s. So this is after Parkinson's time, and that's it today. It's not all that different. He was born in London. He was a son of James Parkinson. His son, in turn, was also named James Parkinson. He grew up in Hoxton Square. He went to medical school in Edinburgh. He was approved as a surgeon, and he was married with 6 children. That's what we know he looks like. We don't know what he looks like, but you will see a picture right there that says that's James Parkinson. That was an erroneous publication. That's a picture of a dentist who had the same name. And the reason we know it's not the real one is James Parkinson died in 1824. And the first daguerreotype didn't come out, the first photograph didn't come out till 15 years after he died. So we know that's not him. There's no portraits of him either. He was a really remarkable guy. Many people don't realize that he was a geologist and known much better as a geologist at that time. He was involved in politics really early on. He was a proponent of universal voting and suffrage. He was an outspoken critic of the government, mainly because they weren't doing enough for the population. And so he was a member of a bunch of political societies, secret ones, where he would publish these tracts under a pseudonym. He didn't publish them under his no name. He actually got arrested for sedition. He didn't get convicted, but some of his fellow members did. As I said, one of the key things about Parkinson's was he was a really keen naturalist and observer. And for any scientist, clinical scientist, basic scientist, the key thing is to be careful in observing. And Parkinson was terrific in observing. He also had one other skill that was really good, and that was he was a whiz at shorthand. So he could take notes. So he could get down his notes and then publish his work. So there was this idea that he was going to try to assassinate the king with a dart, a poison dart. It was called the popgun plot because it was going to come out of this little gun. After that, he started focusing on science more. And he really was involved in lots of science. He was a major author on gout. He published the first paper in English on appendicitis, and it was the first paper ever published that showed appendicitis when you get peritonitis, when the appendix actually bursts. That was with his son. And then, of course, this masterpiece. Okay, so who were his patients? And it ends up there were only 6 of them. His patients, he actually met in his office. He saw 3 patients with this illness that he wrote about in his book, his monograph. One was seen several times. The other 2 were seen once. There were 2 other patients he stopped and talked to in Hoxton Square, right outside his office. And one patient that he writes about in his monograph, he saw across the square, across the grassy area. He didn't even stop and talk to him. And yet out of that, the description of Parkinson's disease is remarkably accurate. And it's a wonderful description for today. So this was case 1, a man rather more than 50. He was a gardener. life of remarkable temperament and sobriety. He started with a unilateral tremor in his left hand, which he initially attributed to being a gardener. He was working too hard. The second case was casually met in the street. Man of 62, he was a professional person. He suffered from the disease for 8 to 10 years, and at this point he had bilateral symptoms, and his speech was very much disrupted, and he was bowed and shaken. So the symptoms, this is Parkinson's description, an involuntary tremulous motion, so they've got a tremor, with lessened muscular power in parts not in action, in other words, at rest, and even when supported, with the propensity to bend the trunk forward and to pass from a walking to a running pace, the senses and intellect being unimpaired. He was, as I said, a remarkably astute observer, but he, even here in his monograph, apologized to the readers that he didn't have enough information and pathological information to really formulate the case as well. So here are the symptoms that James Parkinson really came up with. One was resting tremor, and the other is a slowness of movement, or bradykinesia. He also commented on postural instability and a flexed posture. Notice that there wasn't any comments on rigidity. Many of you may know that Parkinson's patients suffer from rigidity. Well, that actually came in 60 years later with Charcot. Charcot's considered the father of modern neurology. He was also called the Napoleon of the neuroses. Charcot commented and specifically separated out the slowness of movement from rigidity. And he was the one who ended up calling the illness Parkinson's disease. So now we have these symptoms: resting tremor, slowness of movement, rigidity, and often later in the course of the illness, flexed posture and postural instability. And here you can see a picture. Okay, so whom does the illness affect? Well, it's primarily a disease of aging. It happens in older people much more than in young people. There's been a gradual shift towards what we call juvenile Parkinson's, or what was referred to as juvenile Parkinson's. When I started the field, it was 55. Okay, it's now gone below 40, I think, by some people's. And unfortunately, I'm no longer a juvenile according to their criteria. I mean, I had always wanted to remain a juvenile. About 160 out of 100,000 people are affected, slightly more males than females, and there does not seem to be a familial pattern of inheritance in general, and I'll come back to this later. So what's the cause? It's really interesting. Parkinson himself said it really started at the juncture of the lowest part of the brain, the part most in the back, the medulla, the first part, and the transition to the spinal cord. And he said as the disease progresses, the changes in the brain start going more forward, but it never gets up to the front of the brain. The parts of the brain involved in thinking and cognition. He speculated that a toxin might be involved. And that's a really interesting thing, because we now know that there are several toxins that can cause a Parkinsonian-like condition. And London at that time was not a pretty place. It was coal-burning, fires, they were all over the place. The smog and particulate matter in the air was really bad. So it wasn't unusual for him to come up with this idea. So it took a long time for us to figure out where were the changes in the brain. And the first suggestion of the substantia nigra, the black substance, this is an area of the brain in the middle of the brain. It's called in the ventral part, the bottom part of the midbrain. And if you look at this cross-section, it's right here. So it's down, this is the front of the brain, this is the back. It's taken about right here, and this is the substantia nigra. And the first suggestion was on the basis of these authors who found a tumor that caused a Parkinsonian-like condition. But it really took until almost 1920, right after the First World War, for Tretyakov to show that there was a loss of these pigmented cells, and you can get an impression of them right here, and specifically a loss of pigmented cells in Parkinson's disease. So this is what it looks like. So here's the substantia nigra where we peeled away all the material on top, and this is just unstained, nothing's done to it, and right here is the substantia nigra, this area. And you can see there's this black streak. And the black streak are pigmented cells. They have neuromelanin in them. Here is from a very advanced case of Parkinson's disease. And what jumps out is it's very pale. And the reason it's pale is these cells have degenerated. They're gone. So the other thing that came up at this time was that you could spot, if you stain the cells in a certain way, you could spot in the middle of the cell something called an inclusion body. It was a round blob in the middle of the cell, and it was discovered by Louis. It's called the Lewy body, and it's going to figure prominently in how we think about Parkinson's today because of some recent genetic findings. So let's talk about those dopamine cells. Whenever you talk about a cell in the brain, the individual unit of the brain is the neuron or nerve cell. But they don't work in isolation. They work by communicating with other nerve cells and forming pathways which ultimately translate to behaviors. So this is an old from the 1950s. This was obviously the model, the electrical model with electrical wires. I think you can visualize it this way. You have a nerve cell, and it's got a cell body, and these yellow things are called dendrites, but it's got a long process. So it sends out this really long arm, which we call an axon, and at the end of that arm is a specialized area. It does not touch physically the next cell. There's a gap. So how do the cells communicate? Well, the cells communicate by a process called neurotransmission at this region, which is called the synapse. They release a chemical messenger which drifts across this gap, which that gap, incidentally, is about 50 to 100 nanometers. So that's, would you translate that for me? It's very small. It's really small. Okay. That transmitters were first discovered really in the 1920s. But it took until the late 50s and early 60s for first Montague and Carlson to independently define dopamine. And remember that name, dopamine. It's not a dope, it's dopamine. And it's really going to be important for us to discuss. It's the transmitter of those cells that I showed you in the substantia nigra. And Horniakevitz in 1960 showed that you lost dopamine in Parkinson's disease. And then the whole dopamine projections were mapped out shortly later by Annika Dahlström and Kjell Fuchse. So here's a stained section through the substantia nigra, cut in a slightly different plane. And these brown spots, which you can hardly make out here, are dopamine-containing cells. We stain them in a different way here, and you can get an idea why they captivate people. For people like us who are geeks, in many ways, these are wonderful appearing cells. And they've captured the public imagination. So it's in film. This is the movie called Dopamine. And if you could read the subtitle, it says, love, is it real or is it chemical? Because dopamine's involved in a lot of things. This is dopamine tequila. This is one of many albums with the name Dopamine. This one by Mitchell Froom. I would not recommend this album. I mean, it's just not very good. But he's a great producer, and he's got several Grammys for production. Okay, but the album itself, okay. So where do those dopamine cells go? They're in the substantia nigra back in the midbrain, but they actually send those long processes, the axons, up to a part of the forebrain right here called the striatum. And in the striatum, they've got these cells, which are follower cells, and they're called medium spiny neurons. 95% of these cells in this area look like this. Okay, so those are the cells that they're contacting. And what that means is that when you lose dopamine, you have a disorder of striatal, this area up here, getting its input from here. These cells degenerate, you lose the dopamine, so you have striatal dopamine insufficiency as being the proximate cause of Parkinsonism. And this is an image, a PET scan from John Stossel. This is looking down at the brain this way from on top. This shows you the dopamine innervation of the striatum. And here is a fairly advanced patient, and notice how much is lost. And it's a particular pattern that you lose. You lose from the backside first, and it starts sweeping forwards. So this is characteristic. Okay, so what's the treatment? Parkinson's himself discussed treatments briefly. The common treatment at that time was bleeding people with leeches. We do not recommend that today. He argued cogently that it was internal medicines that you would take and swallow, were not justified until there was more knowledge of the disease itself. Interestingly, Parkinson's 200 years ago, he's 1817, he commented on a role for neuroprotection. There appears to be sufficient reason for hoping that some remedial process may be discovered by which the progress of the disease may be stopped. And he tells you why he thinks that's important rather than coming in later. But he says even coming in later may slow progression. So this is a holy grail of Parkinson's disease research. So now you are neuroanatomy experts. You know the substantia nigra, you know the striatum, and now you've got to learn neurochemistry. It's one of these never-ending things. So this is your neurochemistry slide for you to remember for the quiz at the end of the talk. All you got to do is remember, in a way, this part. This is an amino acid, tyrosine. It's derived from your diet. And it's changed or metabolized by an enzyme into a compound called levodopa or L-dopa. And levodopa or L-dopa itself is changed or metabolized into our favorite neurotransmitter, dopamine. So it's a simple biochemical reaction. Your body doesn't ingest dopamine, it makes it, and it makes it from this amino acid. So how do you treat Parkinson's? You give the gift of dopamine. This is another company. I told you dopamine was everywhere. Okay. Give the gift of dopamine. So all we got to do is replace dopamine. That would be the logical way. If you've lost dopamine, you simply need to replace. So here's our pathway. The problem is that if you give dopamine, it doesn't get into the brain. The brain has this wonderful mechanism called the blood-brain barrier that keeps toxins and all out of the brain. It keeps a lot of other things out of the brain as well. And one of the things it keeps out of the brain is dopamine. So there were early studies that gave heroic doses of dopamine to patients that got a slight short-acting effect. But it really took the discovery that you need to give L-dopa itself, the precursor. Because when you give that, it gets right into brain. And then when it gets into brain, it's converted to dopamine. So you replace dopamine by giving levodopa, which many of you know as cinnamon, for example. There's several trade names. And levodopa is also very popular. So here's a picture. This is Maynard Ferguson, the great horn player. David Bowie had an album called L-dopa. There's a company that is a design company. You find this stuff everywhere. All you got to do is look. Okay, so treatment. The treatment now is that we are sufficiently advanced, in contrast to Parkinson's idea, that we do have treatments. Levodopa, or alternatively, drugs that act directly at a protein that dopamine hits, called the dopamine receptor. And you'll hear about these later from Dr. Whitten. I'm not going to discuss these. Which ones are better to use is a clinical decision, and you really need to target it to the patient. The problem with these is that long-term use, after variable periods of time, you have fluctuations in response, and you also get abnormal involuntary movements or dyskinesias. And these can be quite debilitating. So one thing is, we talked about these dopamine neurons, and I want to ask you, how big is a dopamine neuron? Now, to somebody who works in the field of dopamine, it's huge. But if I look at it objectively, that's about 10 micrometers across, 10 millionths of a meter. It's not a very big cell. But you're not really looking at the cell the right way. And you'll see why I'm getting there in a minute. This is a study by a Japanese group that managed to genetically engineer these dopamine cells to express a protein that glows in the dark. It's called GFP. And what they did was, once they had that, they were able to cut the brain into very thin slices, all the way from the substantia nigra to the forebrain. Every section. And then reconstruct. And they found out in a mouse, the average length of a dopamine axon, that process that sends from the nigra to the striatum, is about a half a meter. Now, that doesn't sound like a lot. That's a little less than 20 inches. But remember, a mouse is only about 2 inches long. This is one dopamine cell. But more importantly, the reason it's that long is it branches like crazy. So imagine a tree, a really big, nice oak tree with a really wonderful crown. All those branches come out of a single trunk, the axon. So if you look at a dopamine axon coming into the striatum, it occupies about 2.7% on average of the volume of the striatum. It covers that much. So if you think about that, you would only need 35 cells to cover the entire striatum. That's not a lot of cells. That's what you would guess. And here's some figures. There's an average of 2.7% of striatal volume covered. The maximum was 5.7%. There are about 10,500 of these dopamine cells in the mouse substantia nigra. The striatum has, one estimate is 2.8 million cells. So that means each of the dopamine cells controls 75,000 striatal cells. Now, why is that important? It's great statistics, but so what? Well, the reason it's important is because of this graphical representation, which is a very crude one, incidentally. And what it shows you is that as a function of aging, we all lose dopamine in the striatum. We lose dopamine cells, and we lose dopamine in the striatum. One idea or hypothesis of Parkinson's is that something happens at some point that causes you to lose a cluster of dopamine cells. So now you have this drop, after which you have the normal age-related decline. So that you end up reaching this threshold where you have symptoms. Incidentally, for those of you who do not have Parkinson's, just wait. This data would suggest that when you're about 145, you will have it. Okay, this is a symptomatic threshold. The remarkable thing is that you don't have motor symptoms until you've lost about 70% of your striatal dopamine. That's a huge amount. You've got a minority of the dopamine in the striatum that's left. That has really some wonderful benefits to it. And first is, how do you explain that? And one idea is that you can explain it because you've got one cell in the striatum covering 3% of the volume. There's a lot of other cells that can take over and compensate, up to a certain point. The other thing it tells you is that when you treat Parkinson's disease, you don't have to restore all the dopamine. You just have to restore some of it. And that's a big advantage. So let's talk about what causes Parkinson's. There are data that suggest that it's environmental, And there are clear studies indicating that they're living near a paper pulp mill, rural upbringing with drinking of well water. And the reason for that is it's pesticides. Pesticides get into the water table, you drink the water, you're at increased risk. These are not huge risks, but they're reproducible. So environmental factors can give you a risk. There are also things in the environment that can protect you. Smoking, cigarette smoking, is a reproducible protective effect. The problem is you die many years earlier from lung cancer, so we don't recommend it. Okay, genes. Well, I told you early on that it's not a familial disease. If you try to trace, or if you try to look the classic way at identical twins and non-identical twins, which are the same as siblings, you find that identical twins, if one's got it, that does not predict the other's going to get it. So the idea has long been it's not genetic. But 16 years ago, Mikhail Polymeropoulos and Bob Nussbaum came up with a mutation in a gene, a single point mutation in this one gene called alpha-synuclein that caused an autosomal dominant, a dominantly inherited form of Parkinsonism. Looks almost indistinguishable from what we call idiopathic Parkinson's, which means we don't know what causes it. 16 years later, 22 genes, and this slide is out of date. There are lots of genes that lead to an increased risk for Parkinson's, most of them to an increased risk, and some are dominantly inherited, and the chances are, yes, you will develop a Parkinsonian-like condition. But these genes occur in one family only. So that if you look at the total population, you're still talking about a distinct minority of patients who have any genetic contribution. Okay, so it's estimates range from anywhere from 5 to 15%. So that gene that Polymeropoulos and Nussbaum discovered is called alpha-synuclein. So a gene makes a protein. And in the case of this protein, alpha-synuclein, if you look right here in a rat brain, it's right here in the substantia nigra. And it's really enriched. There are a couple of other genes that make related proteins. This one's beta-synuclein, gamma-synuclein. They're related only in structure. They do totally different things. This is a gamma-synuclein is used clinically to evaluate progression of breast cancer. It's a great marker for that. But it's doesn't have anything to do with dopamine or with Parkinson's. So why is alpha-synuclein important, even though it's in a very small number of families? This is more than one family. Because the Lewy body, which we used to think is only seen in Parkinson's disease, the major protein of the Lewy body is alpha-synuclein. So what does that tell us? Well, scientists dig deeply, and they say, first, what does alpha-synuclein do? Can we learn anything about it? And you're saying, what are birds doing up here? Birds have to learn songs. They have to learn how to sing a particular song, and they learn it from their parents during a very narrow window during development. So the mother sings a song, the young bird sings it back, the mother sings the song back to correct it. No, you got that. That should have been an A sharp minor. Okay, bring it back now. Okay, and that window, right when that window happens of learning songs, there is this huge surge of alpha-synuclein. And as that window ends, alpha-synuclein goes back down. So people think that alpha-synuclein may have something to do with learning and memory. So this is my transition to synuclein and how it's going to tell us about more than just the motor symptoms. So synuclein, as I said, alpha-synuclein is the major protein, the major part of Lewy bodies. And you don't just have Lewy bodies. You have this is a classic, very large Lewy body. Here's one as well with a pale part over here. But those processes, the axons, can get filled up with alpha-synuclein. So they're called Lewy neurites, and you can see that here. This is work from a pathologist named Brock, Heiko Brock, and his wife, the Brocks. And what they did was develop a staging for Parkinson's disease based on the presence of synuclein across the brain, not just in the substantia nigra and striatum. So what they do is they say every case that we have motor symptoms which is defined as Parkinson's disease, and we have loss of dopamine cells in the nigra, they always had these Lewy neurites and Lewy bodies in the dorsal motor nucleus of the vagus. Big name, DMN. It's at the very back of the brain, right where Parkinson's suggested Parkinson's disease starts. And by going back and looking at people who have loss of dopamine cells but no motor symptoms, they also had loss of these cells and synuclein deposits in these cells. So this is stage 1, stage 2, or 3 rather, and stage 6. The nigra doesn't show up as having Lewy neurites until stage 3. So that area that we think of as being critical for Parkinson's disease doesn't even show up till stage 3. On the other hand, this area of the brain shows up very early. So this is the Brock's schematic, and there are 2 areas of the brain that show up before any others with alpha-synuclein. One is the dorsal motor nucleus of the vagus, and the other is the olfactory bulb. Now, why is that interesting? Well, the reason it's interesting is because although Parkinson himself said there was no change in the intellect and there was no involvement of the forebrain, what we now know is that there are lots of non-motor symptoms in Parkinson's disease. So here's a partial list. Olfactory deficits, depression. Depression rates are very high in Parkinson's. Cognitive deficits, dementia, hallucinations, psychosis, impulse control disorders, a lot of different kinds of autonomic symptoms, autonomic nervous system, so delayed gastric emptying, constipation, postural hypotension, which is when you try to get up for example, from a reclining or sitting position. You feel woozy or you might even faint, bladder dysfunction, pain. Some have suggested that the fatigue is really a symptom that is a non-motor symptom. The reason those non-motor symptoms are so important is that when you question large numbers of patients, and they take a questionnaire called a Quality of Life Index, a QOL index, the biggest determinant of quality of life, according to these large surveys, are the non-motor symptoms, not the motor symptoms. That's a really good thing when you think about it, because what it means is that when you go to your neurologist, they can really treat well your motor symptoms. They're doing a remarkable job. Okay. Unfortunately, these non-motor symptoms, which we now know about, are not being treated as well. So this was the original view. Motor symptoms really dominated, and there was no more. Here's a kind of revised view. The reason they're not being treated so well is because they're not based on dopamine. So all of a sudden, where we were focused exclusively on dopamine, we replace the dopamine and it really improves the motor deficits. But it doesn't necessarily, and it doesn't in most cases, improve the non-motor symptoms. So that's the next frontier. But I think the ability to define what's the underlying changes give us hope that we can indeed treat them. So if you look at correlation, olfactory bulb right here and the dorsal motor nucleus right here are the first areas to go. This would say that you should have olfactory bulb involvement, you should have olfactory symptoms. You should have a deficit in smell. And many Parkinson's patients do indeed suffer from deficit in smell. The maximal, if you take an odorant, something that smells like, imagine mustard, okay, if it's maximal concentration, they can get it, but they're not as sensitive. Okay? the dorsal motor nucleus controls things like heart rate and blood pressure, what we call orthostatic hypotension, when people fall when they're rising up rapidly, okay? And all these autonomic symptoms. And you can start relating and saying, well, depression should be up in the cortex, whatever. This gives us hope that we can define and start targeting these areas to define new treatments for non-motor symptoms. So that's the end, and I was being waved off by Dr. Ingram kindly, gently. Finish up.